This is Mr. Maxwell from Pingree AP Biology, and I want to tell you a little bit about some research that I did about 15 or so years ago when I was working at the University of Wisconsin for my master's degree. Um, did this work in between 1992 and 95 and published it in 97. Title of the paper is Effects of Water Stress on Colonization of Poplar Stems and Excised Leaf Discs by Septoria musiva. So what the heck does that mean? The question really is, do thirsty trees get sicker more? So is it more likely that a thirsty tree is going to get sick? And first I need to give you a little bit of background about hybrid poplar. And the question I need to ask you is, what are these? And the answer is, well, they're sticks, but they're kind of cool sticks. Each of these is a different clone of a poplar hybrid. And basically what you can do is you can take these sticks in the winter off of trees, and if you put them in the ground like this, in one season, they'll look like this. And so hybrid poplar has a really fast growth rate, and this is why people were interested in looking at them for a biofuel. So basically the idea is you grow these plants up couple years, and then you can harvest them. This plantation here is about 5 years old. If you just want to get fuel, you can grow them up for 5 years, and then they're chipping them, and then they'll use the chips to power a turbine to generate electricity. You can also leave them for another couple of years and use them for lumber, but we were mostly interested in hybrid poplar as a biofuel. So that's why they're important. Hybrid poplar tends to get a couple of diseases, one of which is called Septoria musiva, and it causes a leaf spot and a canker disease. And the question, if this was alive, that I would ask you is, why is a leaf spot bad for the tree? And you should be able to see here that a leaf that's damaged by a leaf spot doesn't have as much photosynthetic area, and so isn't as efficient at trapping light, in addition to the fact that it's being eaten by a fungus. And then if you look at the stems here, the cankers, these are cankered tree limbs, and these two are the controls. But you can see that, any vascular tissue here, the tree has to move water and nutrients from one place to another through its stems. And if there's, big lesion here, that makes it difficult for that to happen. And then if the canker is located on the main stem of the tree, the question I would ask is, why would this be bad for the tree? And you should be able to come up with, if you think about it, obviously there's difficulty in transporting nutrients from the roots up to the leaves, and sugar from the leaves down to the roots. But this also weakens the stem and makes this more likely to happen. And of course, this tree is not gaining biomass, so it's not a very good condition for a tree to be in. So the plant is pretty the disease is pretty bad for the plant. The next piece I got to tell you about is drought stress. And to understand how a tree gets stressed because of lack of water, you got to think about, how does water get up to the top of the leaves from the soil, which in this picture we're basically lying on the soil looking up. And so the water has to move from the ground up the tree. And the simple answer is that as long as the soil is wetter than the air, water will move to where it's dry. Later in the course we'll talk about water potential, and we'll do some calculations and things, but at this point, if you understand that water moves from the soil into the atmosphere through the tree because the air is drier, that's really the force that drives it. So if you think about it, every leaf on this tree is pulling on a continuous column of water down into the soil. And so you can think of it as the leaves are sucking on a column of water. And if you want to know how strongly they're pulling on that column of water, what you can do is you can put them in a device like this called a pressure bomb. And basically what you do here is you put a leaf in the chamber and put it so that the petiole is sticking out. And so this leaf would be drawing water from the stem into the leaf, or the petiole into the leaf. And what you do then is you put compressed air in this chamber, and that will push the water out of the leaf. And when you see it just make this top of this petiole wet, when it just becomes moist, the pressure that you apply here is the same as the amount of pressure that the leaf is exerting on the column of water. And so this is a real good way to measure how strongly the leaves are pulling on the water. And trees that are under a drought stress have to pull harder to get the water up. And so the standard for measuring water stress is to take leaf water potentials right before dawn, before the sun comes up. And that gives you a really good read of how strongly the leaves are pulling on that column of water when there's no sunlight affecting it. So you got to get up before dawn and take those measurements. Now, you also need to know about how we inoculated the trees. Inoculation means applying a fungus in this case. So give you a little bit about the setup. We had two clones of the tree, NM6 and NE308, and we grew them in a common soil environment so that NM6 and NE308 would be subjected to the same amount of water in their soil. We had two watering setups. Once we watered 3 times a week, Monday, Wednesday, Friday, and then the other ones we only watered them after they hit a significant drought stress. So don't water them, don't water them, don't water them until they hit a significant drought stress, and then water them. And then we also had 2 inoculation treatments. One was just you pull a leaf off the tree to make a wound, and you add poplar leaf decoction agar. And the other one, you do the same thing, pull a leaf off to create a wound, but then the poplar leaf decoction agar also has a strain of the fungus on it. And so one question, if this was class, I would ask you is, what is this treatment here, the wounding? And hopefully you would be able to tell me that it is the control. Mostly what I want you to get out of this is not what all did Mr. Maxwell do for his master's degree, but rather experimental design things. I think it's a reasonably well-designed experiment. Okay, so here's the setup. Each one of these boxes had both clones in it, a number of individuals of the same clone, and there were 30 of these boxes set up in the greenhouse. You see the trees are small. By the time the experiment's over, they're pretty tall. This is just as we set them up. And then this is a graph that shows on the y-axis here pre-dawn water potential. So 0 water potential, 0 megapascals would be pure water sitting in a container open to the atmosphere with nothing in it, no solutes. As you go more and more negative, this is drier and drier and drier conditions. And then on the x-axis here, you see day of the year. So this is pre-dawn water potential over time. And you'll see here that we have NE308 well watered and NE308 stressed. So the watered condition would be the circle, and the stress condition would be the square, And if it's not colored in, it's NE308. And here we have NM6 watered and NM6 stressed. And basically what we did was measured a couple of leaves on trees for a while, and then when we noticed that water potential was going down, and by we I mean I in this case, until we got we started seeing a separation, or thinking that we saw a separation. And then we monitored a little more intensely. I took 6 leaves off of each type of clone and measured them for this day. And then also same on this day. But then on this day here, we had, I got everybody in the lab to come in, and we ran through 60 trees for each treatment. So, you know, it was an all hands on deck thing. You got to pull all these leaves off and do them all before the sun rises. And so for these 3 days, we intensively sampled. And my major professor, when we were designing this experiment, when I presented the experiment, he wanted to know, why are you doing so few samples? And I answered, well, because that's how many I can do. And then he said, well, we really need to get that sample size higher. And this is how we came up with the idea that we just get everybody in there to do it and do the all hands on deck thing. Because, the question of how many samples should I do, more samples is always better because you get more statistical power out of it. And you can see here that on these 3 dates, these numbers are significantly, the ones that we withheld water from are significantly below the other ones. You can also pull some other things out. You can see that even though these guys were in the same soil environment, that they were not subjected to exactly the same drought stress. You do the best you can do, right? So then what we measured as an outcome was first the canker length, and then how much of the stem was girdled. So canker length is basically you take a ruler and measure how large the canker is in length. And over here you can see the results. Here we have canker length on the y-axis, and then on the x-axis you're going to have this data, these labels down here apply to this as well. So these 4 bars over here are for NE308, and then these 4 bars on this side are for NM6. The open bars, the ones that are not colored in, are the controls, and then the darkened ones are the inoculated ones. And if we were in class, I'd have you interpret this, and we'd come to a class conclusion. But what you should be able to see here is there are a couple things going on. First of all, did the inoculation work? Yes. We got more disease when we put the fungus on, bigger cankers when we put the fungus on than when we didn't, which is totally expected. The fact that we saw some canker when there wasn't any fungus on, it's probably not really a canker, it's probably just a wound response. And then you can also see that NM6, regardless of condition, had smaller cankers than NE308. And that's what we expected because NM6 was the supposedly resistant clone, and NE308 is supposedly more susceptible. And then we also saw an effect of water. So well-watered trees had less damage than stressed trees, and that was true for both of the clones. We also looked at how far around the stem the fungus grew. It's called percent girdle, and basically you just kind of estimate how far around the stem the fungus grew. And so once again, here we have percent girdling on the y-axis, and then we have for NE308 these 4 bars, and for NM6 these 4 bars, and again the control and the inoculated. And so for the inoculated NM6, you can see that well-watered trees had smaller cankers than drought-stressed trees. But we didn't see the same thing in any 308. And there are a number of reasons why this might be, and you should probably, if we were in class, we'd discuss why, but there are a number of reasons why this might be. Okay, just to review the experimental setup for this next experiment was basically the same. We used the same trees, but then we cut leaf discs from leaves from each type of tree. So there are basically two inoculation treatments in this. We used sterile distilled water, which is the control, of course, and then we put Septoria musiva canidia. These are spores. In distilled water on the leaves, and then use a computer to measure how much necrosis, or how much area of black there was on the leaf. And so, here's a sample of one of the trays, and you can see this one has very little necrosis, and this one has some blackened areas here. And this is the computer we used to do it, and basically this camera took an image of each one of these leaf discs, and I would click, 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 take a picture of this one, click, 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 take a picture of this one, click, 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 take a picture of this one, and then I had the computer tell us how many pixels in the image were below a certain darkness threshold, how many were black, and then how many were green, and then it estimated percent necrosis. Back in the 90s, this was kind of relatively new technology. We actually had to buy a product that was a computer product that was just declassified from the military to be able to do this. Nowadays, there's actually a free program that you can get on the internet called ImageJ that does this stuff. Same type of thing, but it's amazing how far things have come. So anyway, here's our data, and this is set up the same way as the previous graphs. You can see NM6 is over here, NE308 is over here. Percent necrosis is the response variable. It's on the y-axis, and this first graph is for after 1 week of incubation, and then B is after 2 weeks, C is after 3 weeks, and D is the same discs after 4 weeks. And you should be able to see that a couple things. You pause the presentation and figure it out for yourself, but you should be able to see that NM6 pretty much all the way through had more necrosis than NE308. You should also notice that the well-watered plants had more necrosis than the stressed plants, and this was true for both clones. While it wasn't significant in some places, it was significant in a lot of the times. So now, if you think back to the to the other data I showed you, to summarize this, leaf discs from well-watered trees had more disease than discs from drought-stressed trees, and you look at the data I showed you before, you'll notice that this is the exact opposite of the result that we got with the canker length. And when my major professor first saw this, he was convinced that I had mislabeled the axes because this should have matched this. But it didn't, and I didn't mess up the axes. So then you have to speculate on, if that's the real data, if that's what actually happened, then you have to come up with reasons for, why do you see a difference in the one assay versus the other assay? And if we were in class, we would discuss that. But I think, you can come up with reasons. So the conclusions from this, at least in intact trees, drought stress exacerbates stem cankers. In the excised leaf disc assay, which is a little bit artificial, discs from well-watered trees wound up getting infected and colonized faster. So A follow-up question for this then is, of course, is that true if the leaf's still on the tree, or is it a factor of it was on the tree and you took it off, and it's going to act a little bit differently when it's off the tree? Um, this effect might be drought stress trees have have more closed stomata. So if the fungus is getting in through the stomata, it might be harder if the stomata are closed. Drought stress trees also produce leaves with fewer stomata. So it might be that there are fewer places for it to get in. Could be the nutritional status of the leaf. It could be a lot of different things. And good research, you always wind up with more questions than you started